Here we are with AP Biology Experimental Design and Statistics lecture. This would be the second one in this topic. We've previously talked about the independent variables. Let's go through this quickly, okay, at some point. We talked about the basic format of the scientific method that we'll use, the hypothesis, defining the independent variable, okay, and yeah. We learned that a hypothesis is not really an educated guess. We learned it's based on a proposed explanation based on limited evidence that we can set up and it's testable, okay? And we went through the theory and the law, all of which you can read. It's not the purpose of this demonstration or this lecture. And a hypothesis almost never becomes a theory, but could be, and theories don't become laws. And again, we've covered this once before. Theories are basically explanations, and whereas laws are basically observations using a lot of times formulas. Okay, so we can go through that. And we define the independent variable. We've done it before, but we do so understanding there's two types of properties. There's qualitative properties, properties that are not based on a number, they're based on a quality. For instance, I smell, or there's water has lead in it. Okay, quantitative properties, as we talked about, are something that we can measure. You know, what is the actual temperature as opposed to saying it's warm? Mr. Grodsky's odor temporarily paralyzed 30 ducks in a controlled experiment. That's a number that we can measure. The water has 7 parts per million of lead in it. It doesn't just tell me it has lead, it tells me exactly. So these have numeric values that are tied to some unit that we can measure. So quantitative versus qualitative. And we're mostly going to deal with quantitative properties as we go through our experiments. We talked about the dependent variable usually being the y-axis. We want to measure how much it depends or changes based on changes we make to the independent variable, all of which we've covered before. And we've talked about the independent variable at length, so we're going to get through this, okay, and continue on our discussion at some point. So now we're on data analysis and statistical analysis, and this is where I really want to spend my time on. So we're looking at some information. Let's look at a scientist typically collect data on a sample of a population, and that's important. This is really important. We can never really take information on an entire population. That would be very, very costly, but we can deal with samples. That's very important. We've got to make sure our samples are representative of the population, and this is called, we don't want to stay away from sampling error. So we want to use a sample of the population to study and to use the data. As long as our sample is representative of the population, we can draw a conclusion or make inferences about the entire population. An example of a data set is shown in Table 1. It shows beak measurements taken from 2 groups of medium ground finches that lived on an island of Daphne Major, one of the Galapagos Islands, during a major drought in '77. Now, the Galapagos Islands had finches, and what's interesting about those islands, they were separated from South America. So they really are their entire ecosystem, and it's really cool to study those, and they're studied often because evolution occurs at these very, very isolated places, meaning there's not a lot of influx of other genes to make our experiments change. In any case, we noticed that there was a major drought in 1977, and we know that one group of finches kind of died off, and those were the ones with smaller beaks. So we can look at this information and measure the body mass, and these are band numbers that identify the birds, and their wing length and tarsus length and their beak depth. We're after the beak depth, and you can look at all this information, and we gathered all this information. As you can see, how helpful is just gathering information. You can't really draw much from just looking at these numbers, okay? But if we were to take this information and put it in a chart like this, a very simple bar graph, we can see the number of birds, okay, depends that we measured in groups at beak length. And we can see that an average of the beak length of the birds that did not survive the drought because their beaks were too small was 9.1 milliliters. The next page, I have the average 9.67. So if you remember, if we go back, we can see that the average of all of the birds that we looked at, we didn't look at the entire population, it was a sample. And we noticed that that sample, we get an average, which is called a mean of 9.11. And that's for the what? the birds that did not survive. When we go forward, we can see the birds that did survive had bigger beaks. And what we think is very clear is that the birds that had the bigger beaks were able to get at the harder, drier, bigger nuts. Remember, there was a drought. There was really competition for food, and there was only so much. The ones with bigger beaks had the adaptation to survive the environment that changed. And we had a die-off of birds that had smaller beaks. But the bottom line here is I'm after central tendency. Notice if we're getting a mean that our information would spread across. So we can see that there's a general spread of birds. And we would say that this is showing a central tendency because it looks to me that the sample is indicative of what we'd see in nature. You're going to see some things that are really, really small and not many of them. And you're going to see some things very, very big but not many of them. Most of the animals will have beaks close to the average, around the central mean. So that's why we look at this. So we're graphing this and we're seeing the spread of data. And the mean for the birds that survived had a larger beak. They were able to use a larger beak to break open the harder seeds that the little beaks could not. And that's why they survived. This is an example of natural selection. Okay, so as we go forward and look at the data, we can see that the measurements tend to be more or less symmetrically distributed across this range, with most measurements around the central or the center of the distribution. That's what we say a normal distribution. We would expect a normal distribution in the population, so our samples that we're taking should also indicate that. Remember, the samples are what we take in a lab or an investigation to hopefully get what the actual distribution is. Okay, and this type of statistics, looking at the central tendencies, is what we're after today. So you can see that there's a shift in this central tendency, whereas the mean is 9.1, here it's 9.67. It's shifted to the right, okay, in this type of natural selection. So the graphs help us look at these central tendencies, okay. So how do you describe these two graphs? How are they same or different? Okay, graphing helps us have a visual, but we need to do some descriptive statistics in order to actually get some values that show us that there's some quantitative differences. Okay, so what is statistics? What is descriptive statistics? Easy for me to say. It's a branch of mathematics that provides techniques to analyze whether or not your data is significant, meaningful. We'll get to that. Statistical applications are based on probability statements. Okay, we've seen that in our Punnett squares and our Mendelian genetics. Nothing is proven with statistics, right? We can only say that something may be occurring. There may be a link. We can never say definite, okay? Statistics are reported. And statistics report the probability that similar results would occur if you repeated the experiments. Reliability issues. So these are very important statements regarding the value of the numbers that we get from investigations or labs that help us find some meaning. What do these numbers mean? That's what we're constantly after. We can't just look at the numbers or a graph and say what they mean. We have to have some more definite, hopefully some proofing here that gives us a better, firm understanding of what the investigation said. And statistics help us do that. Now, of course, statistics deal with numbers. It is quantitative, okay? And it's important that you understand there's two types of quantitative or numbers or quantity. They're continuous variables. These are numbers associated with measuring or weighing or any continuous interval, meaning when you have a weight of something, a weight could be 11.4 pounds, 11.44 pounds, 11.443 pounds. It could be any number. It's a continuous interval, all right? Whereas like time, it could be 3 seconds, 3.5 seconds, 3.55 seconds. These are continuous variables. Then we have quantitative values that are discrete actual numbers. These are when you like categorize something and count how many stems, how many flowers on a plant, how many trichomes, how many boys, how many insects, a definite number. And these are discrete variables. And we will basically be focusing on discrete variables in this course, okay? And from these discrete variables, we will be able to look at some values of the average, mean, median, mode. So in these two graphs, we can see that there is a central tendency, but these words, the mean, median, mode, are type of descriptive statistics that help us understand these central tendencies. Okay, now the mean. Okay, very simply, the mean is an average. Now, the average of the sample, this little X with a line over the top, really means the average of the sample. And now, of course, we're hoping the sample that we took in our research was indicative of the entire population. Okay, so what we do is we sum up all of the X's, all of the values that we collected, and we divide by the number in my sample. And that should, if I have a what? Random sample that representative of the true population, will give me a mean that represents something of the population. Okay? So in any case, you're done with averages. Add up the total number that you have and divide by how many pieces you have, or n in this case. Okay? So for instance, well, in the case, samples are selection of the population which are used to make inferences about large populations. This is important. So these averages that we're getting help us make inferences or understanding about the large population. We can't find the entire population, the real population, because it's just too costly and time consuming, but we can use samples. That's very important you get that. So individuals in a sample population must be a fair representation of the entire population. Right, you have to pick samples that are random sampling, so that you get a true understanding of what the population is. Therefore, sample members must be randomly selected to avoid bias. Example, if you're looking for the strength or how much people can, let's say, bench press, or how many push-ups people can do, and you're looking at strength in students, you wouldn't pick students from the football team. They tend to have more physical attributes in strength than other students, let's say, since football is due to pushing and pulling of other people around. So that would be a bias. You take a random sample of students, not from the football team, who are already pretty good at being strong. That's why they were selected for the team. Other examples here. Let's think of a cage rat has a cage has 1,000 rats. Okay, you pick up the first 20 so you can catch your experiment. You pick up the first 20 you can catch for your experiment. Well, I can read today. So think about that. I'm doing an experiment with rats, okay, and it has 1,000 rats. It's pretty nasty, but the first 20 that I'm able to catch I use for my experiment. Well, maybe the ones that I just picked were not a random sample. These were the slowest. So if I had some kind of movement study or study that involved anything, I picked the slowest and sluggish of the rats. So that has bias. A public opinion poll is conducted using telephone directory. Are the people listed in directory have some kind of bias? Yeah, if you're located in a telephone directory, maybe the people who look in telephone directories have the ability to read, okay? Or maybe they're more educated, who knows? But if you're just using telephone directory, you're dealing with only a small section of population, okay? 3, you're conducting a study of a new diabetes drug. You advertise for participants in a TV or newspaper. So is that, you advertise for people only in what? Newspaper and TV. So you're only getting the people that read newspaper and TV. Now you already have a select part of society that you're picking from. That's not a random sample. People in a telephone directory only select part of society. The 20 you can catch is a select part of that society. These all have bias. Rats you grab are slower. Telephone you call only people who have a phone. Maybe they have wealth. And people who are listed, maybe they're responsible. Newspaper, TV, you reach only people with newspaper, wealth, educated. So the point is that we're not getting random samples for my studies if I pick samples that are not representative of the entire population. These all have bias, and these would have sampling errors. Now, if you use a sample population, meaning you have a sample that represents the entire population, you can get an estimate of the actual mean or the average of that population. Notice if my numbers 1, 2, 3, 4, and 5, the average of them, if I add them together and divide by the 5 numbers, that just means sum up the individual numbers, divide by how many you have. I get my average to be 3. Notice the mean shows that half the members of the population fall on either side of the estimated value. Why? Because we have a normal distribution, normal curve. So I have a study in a biology class. I have planted 8 bean seeds in separate plastic cups and placed them under a bank of fluorescent lights. 14 days later, the students measured the height of the bean plants that grew from these seeds. All right, let's find the mean. We add up all of my values. Divide by how many I have. 1, 2, 3, 4, 5, 6, 7, 8. What do I have? 8.7 centimeters. And that is my mean. Now, is that the true mean of my population? No, it's an estimate. And hopefully, okay, if I did my experiment carefully enough, okay, I would approximate that. Now, however, 8 bean plants does not give me enough variability. If I had 8,000, we hope that this number would be more, would be closer to the actual. But it's a good estimate, all right? Median, you should be aware, is different than the mean. Okay, the median is the middle number. So if you have a, let's say, time it takes for mice to locate food in a maze, these are a little different times. What you do is essentially you order them from lowest to highest and find the middle number, and 32 is my median. Okay? Now you may say, well, why would I look at the median if I could look at the mean? Well, sometimes the median is a better middle number than the mean. For instance, if I look at the mean, which is the average, is 41 seconds. And the reason why it's so much higher than the middle number is because one of your values is really, really, really big. So we had a slow mice. One of the mice that we studied was really, really, really slow. And that little aberration, maybe it was sick, made the rest of your data skewed toward 41 seconds. It would have been a lot lower if you didn't have this one really, really slow mouse. So the point being is that the median sometimes give you a better reflection of the central tendency if one of your values is outrageous. Okay? Now the mode we don't often use, but the mode is where you have the most numbers of the same event. So in this case, for this case 33 was the mode because that was the one that was one of the values that repeated the most. And sometimes a mode can help us if we don't have a central tendency. Notice there's two central tendencies here. So having a mode will give us a little more insight of what's going on with the data. Okay, now measures of variability. This is really getting to the most important pieces of this. We have range, standard deviation, and variance. Variability is very important. It describes the extent at which numbers in a data set diverge from central tendency. It's a measure of how spread out the data are from the mean. That's important. Okay, let me tell you what I'm talking about here. These two distributions of test scores in people both have the same exact mean, average. So we both have the same average here. But what's different about this? Well, where's the variability? Even though we have an average of the same 150 test score, whatever that means, you can see there's a lot more variability from that number than it does here. So how spread apart the rest of the data could be very, very important in terms of biology. Frequency, okay, or maybe predicting what is normal. If you're within a certain value, maybe that's normal. So variability really talks about how spread out the numbers are from that central tendency. You can have the same mean, but you can have a lot of people close to each other or very spread apart. So variability is very important to look at. One of the first is range. What you do for range, very simply, is take the largest value of a data set, in this case 10.3, and take your smallest, 5.7, and then subtract them, and your range is 4.6. And that can give you a measure of variability. Okay? Now, if you look at variability, though, in terms of range, there is a problem here. If you have a very large or small variable in the data set, like we talked about that slow mouse, that can really disrupt your understanding of variability. For example, if the maple leaf sample had not included the very small leaf number 5, okay, the range would have only been 2.8 milliliters. What I'm trying to say here is that if I take out one of my really small values, okay, the range would change a lot. So sometimes one of your data points could be off so much to disrupt and give you an idea of the variability that's not really true, just like we talked about in the median, or mean versus median if you've got a number like the slow mouse. So this 5.7 makes my variability greater than it really is. Okay, and it's an aberration because it's much smaller than the others. So we need a better variable, and variance is it. Okay, now how do we get a variance? It's given s squared. This gives me the degree of variation, and this is where it gets a little bit tricky, so pay attention. A large variance means that the individual scores of data are really spread out from the mean. A small variance means they're close to the mean. And this is a better value than range because it takes in consideration those weird small or big numbers we talked about. Okay, so how do we do this? Okay, well, what we do is we take our numbers, okay, our measured values, and we have a mean. Okay, the mean here for these values is 8. Okay, and here's what we're going to do. We're going to subtract our value from the mean. So 10, which is our numbered value, minus the mean. Okay, and what we do then, so we keep subtracting our values from the mean. So the mean is 8, so 10 minus 8 is 2, 7 minus 8 is negative 1, 6 minus 8 is negative 2, 8 minus 8 is 0, and 9 minus 8 is 1. This x1 is our measured value, x is our mean. So all I did here is take this value and I subtract it from 8, which is the mean. You had to know that. I'm telling you that. Okay, now, then take the value. So 10 minus 8 is 2. Take this value and now you square it, and you get 4. What the squaring will do is take away those negatives, okay, and make them positive, okay? And also will mess around or take away and flatten out those arbitrary data points that are too big or too small. It will definitely help with that. All right, so then we basically subtract them, and so we take the value minus the mean, you get this value, and then you square it, and then you add these values together, and that's how you come up with the variance. Now we're not done yet, but we'll get there. And then you sum all of these values. So we take 10 minus 8 is 2, 7 minus 8 is negative 1, 6 minus 8, negative 2, 8 minus 8 is 0, 9 minus 8 is 1. Take these values and square it. Okay, just square them. And then you add these up, and you get your 10. We're almost there. And then we take our 10, which is what this equals. Kind of weird, but you sum of, okay, and you divide by what we call degrees of freedom. Since you have 1, 2, 3, 4, 5 different values, you have 4 degrees of freedom. What you do is you minus 1. Now the reason we do that is because if you have 4 values and you know what they are, you would know what the 5th one is. And I can explain that a little differently, but the bottom line is, if you have 5 values and you know 4 of their values, you can figure out the 5th. So that must mean you only have 4 numbers that can wiggle and change. I'll say that again. If you know you have 5 values, and you know what they add up to in this case, okay? And if I know they add up to, let's say, 40, and I have a 10, a 7, a 6, and an 8, this has to be a 9 to equal 40. There's no wiggle room, so this is not a degree of freedom. These can all be different numbers, but once I get to the 4th, the last number has to be a number that adds up to 40. So what you do is you basically take 1 away. So if he has 5 values or 5 samples, we subtract 1 to get degrees of freedom. And then we take our value that we got from the last page, the 10 that we got from adding up, that's the sum value, and that was the squares that we added up, and we divide by, it was 5 plants, we minus 1, and 10 divided by 4 is 2.5 is the variance. And this is one good way of looking at the degree at which individual members within a sample vary from the mean. So we got this value that we added up here, and then we subtracted the degrees of freedom. There was 5 plants, so there's only 4 free numbers we can make. Okay, and then we get this value here, variance. Last thing I'm going to do, I know I talked too long, standard deviation. Now, variance was s squared. Okay, so if you notice, this is s squared. This standard deviation is s. So what we're going to do is just take the square root of s. So we just found this. Taking the square root of s gives me a value called standard deviation. So we take our 2.5, the square root of that gives me 1.6. Now the powerful part of this, party people, is that the standard deviation can give me something I can predict with. Let me explain. Here's a standard distribution. Here's my 8 centimeters, which was my average. One standard deviation goes from 6.4 to 9.2. How do I know? It's plus or minus 1.6. So I add my standard deviation I just got from my other page is 1.6. It was the square root of the variance. Okay? Now, this standard deviation 1.6 means add it to 8, you get 9.16. Take it away from 8, you get 6.4. So this is called plus 1 standard deviation. This is 1 standard deviation. So between plus 1 and 1, we call that 1 standard deviation, right? And we say 68% of my normal distributed curve fits in this area. So we can predict that 68% of the population will have heights between 6.4 and 9.6 within 1 standard deviation, plus or minus. And we can say 95% of all the heights in a population will be within 2 standard deviations, plus or minus 2. And we can say what? The probability of finding a p-plant above 12.8 or below 3.2 in the absolute crazy what? Parts of our curve, our outer edges, is less than 1%. So knowing the standard deviation will tell us how normal we are. If I find something, my average is 8, and the standard deviation is 1.6, pretty small, I can say with pretty good confidence that I have a good normal number. If my average is 13, it's not within that bell curve. I know that I've got some p-plant that's really, really out of the norm. These p-plants can exist in this range as a normal population. That's what we're saying. And we can predict based on standard deviation how much of that can exist. Case in point, if you wish to see if a red blood cell count was normal, you could see whether if it was within 2 SDs of the mean as a whole. So within 2 SDs, you can see whether or not you have a normal red blood cell count population. What's normal? Well, we take measurements from a sample population and we get a mean. Then we take the standard deviation and see how spread apart people are. So when you get back a value of your red blood cell count, if you did blood work, we know if you're normal. If you get something in this area here, there's a problem. And we don't just make up those lines, we use it based on statistics. So if you fall within 2 standard deviations of the mean, that means 95% of the population is covered. If you're out in this area, there's probably something occurring. So what I want you to do is go to the worksheet, using the formulas given to you, try to come up with the mean, the mode, the median, the variance, the range, and the standard deviation. If you have to go back to this tutorial or these slides, which are right in your website, right in the website webpage, go ahead.